All right, welcome everybody. So we're meeting tonight. I'm actually recording this just before class, but one of the things I want to talk about next week for the next lab is t-tests and Anovas. I don't know if we'll get to that tonight so much, but I want to put it on record it so that you can access it later on and watch it before next week. So that's what we're going to cover is t-tests and Anova. So let me go ahead and share that with you. All right, t-tests and one-way Anova. So that's what we're looking at here. These are both designs where you're manipulating an independent variable. So a classic experiment, you're either having one independent variable or you're having one independent variable with two levels or one independent variable with more than two or more levels with the one-way Anova. We're going to talk about each one of those. Here's examples in a second, but it's some of the basic design stuff that you'll have within a research study. So t-test one-way Anova. Let me jump in here. So whenever you are completing a research study, whenever you are manipulating a variable, whenever you are doing an experiment, that's what we're talking about. Last lab, we were dealing with correlations and regressions and chi-squares. So not exactly experimental stuff, but more collecting data and information and then correlating it. This week, or for this lab, we're going to look at experiments. So an experiment specifically done to assess causality. Remember, to establish causality, you need to do an experiment. So here, and let me grab a pointer. So here, we are looking at the independent and dependent variable. Okay, so the independent variable is manipulated or controlled. The dependent variable is measured. By manipulated or controlled, it's the variable that the researcher is in control of. The variable that is then used to evaluate whether there's a difference is the measured variable. You can have multiple ones or singular ones. And we use t-tests and Anovas to determine the impact of an independent variable on a dependent variable. And each of these designs have multiple ways in which they can be set up. And we're going to look at some of those examples, but all of them are analyzed using a couple of different statistics. So we will use t-tests and Anovas to determine the impact of the independent variable on the dependent variable. That's what the technique we're going to use. So one of the designs is you may want to compare two separate groups. So we have here a control group and we have a cognitive behavioral therapy group, right? So a CBT group. And we might want to compare if this group here is different than the cognitive behavior group. This can be done using an independent t-test, where one of the groups is a control group, one of the groups is an experimental group, and you're seeing, are there differences in a whole manner between this group and this group, okay? All right, sorry about that. I had a phone call. So we're talking about independent, dependent variables. And if you have the previous one, we were independent t-test. So you're looking at two different groups, and we're going to define what independent t-test here is in a second. One of those groups is a control group, and one of them is the cognitive behavior group. And you're trying to see, is there a difference in this in these individuals based on this therapy? So that control group might be someone who has a particular issue that needs resolution through cognitive behavior therapy. You do a control group on that in those individuals, then you would assign another group to receive therapy, and then you compare to see if there's a difference in the resolution process of those individuals. Another one, independent t-tests, you could do two separate groups. Say you want to compare two different types of therapy, art music therapy and cognitive behavioral therapy. Which one works? Again, you have different individuals. So that's what person one, person two, person three means, is you have different people in each of these conditions. And then the cognitive behavior therapy, you're comparing it with 5 other different people, separate. That's why they're independent. They're not related. They're completely separate, independent groups. You could also have a dependent t-test. And in a dependent t-test, you have the same group under 2 different conditions, and then you can compare them. So you go back to that control group and the cognitive behavior group. Your control group is your same people that are receiving the therapy. So it's kind of a pre-post. You have a control group, which is the pre-situation. You measure them. You then administer the cognitive behavioral therapy. and then you assess them again at the end, and you see if there's a difference in the change in that individual pre and post. So those are t-tests, right? You're just comparing one independent variable, type of therapy, and two different levels of that independent variable. So two levels of that independent variable. But that's not the only thing that you can look at. Sometimes you might want to look at 3 different groups or 4 different groups. So what can you do? Theoretically, you could do a t-test and do control group versus cognitive group, control group versus art therapy, control group versus traditional therapy. There's a problem with doing that statistically, is that theoretically you add more error every time you do another comparison. So it's better to do an overall test, and that's what an Anova is. And so in a one-way Anova, you compare more than 2 levels in an independent t-test design. So you have a control group, you have 5 independent people. You have a cognitive behavioral group, which is another 5 people. You have an art therapy or art music therapy group, which is another 5 people. And then you maybe have a traditional therapy group, which is another 5 people. And then you're comparing them across all of those groups. This is a really basic way to analyze one independent variable, right? The independent variable in this condition is still therapy. The independent group in this independent variable in this situation is also therapy, control group or not. Therapy and therapy. What varies is the number of levels of that independent group. Okay, so let's introduce the t-test. The t-test is a way to analyze, like I said, and compare 2 sample means. We can use standard scores or z-scores to compare a sample mean to a population. We don't usually have population information. Instead, what we usually have are 2 sample means. So if we have 2 sample means, how do we do that? How do we make those comparisons? Well, we can then compare mean 1 to mean 2 using a t-test. So let's look at an example. So a researcher is interested in determining whether there are differences between men and women voters in their attitudes towards universal health care. And so to do this, random samples of both men and women are obtained. They're given an attitude scale on health care, and then the means for the 2 samples were computed. So we have a mean for women here of 40, a mean here for men of 35. The researcher then becomes interested in determining, is there a significant difference between men and women in terms of this attitude? One possibility is our null hypothesis. Remember, our null hypothesis states that there is no true difference between the men and the women, and rather the only reason that there is a difference is because of random sampling error. Okay? In this example, we're using 2 samples obtained from a survey in order to draw inferences to relevant populations. So think of it this way. The null hypothesis states that men and women come from the same condition. So the men are 35, the women are 40, they're from the same group. Men just happen to be on the smaller side, women on the higher side of that distribution. Okay, that is one population men and women are part of. The research hypothesis actually states that the population of men and women are actually different, and the mean of men of 35 is here, the mean of women is 40 of there, and those populations are different. And the reason that they're different is because they have different beliefs, right? So in this situation here, null hypothesis, same population. Here, separate populations, research hypothesis. There's a couple more examples. I'm going to let you guys read through these. There's an example 2, kind of goes over the same thing, but I don't want to spend a lot of time on those. I think you can kind of go through those. I hope you get the idea of what I'm talking about here. One deals with vitamins and placebos, and then I guess that's the only other example I have there. Both experiments and surveys are frequently conducted within in psychology, and in both cases, what we usually obtain is sample means. That's what we're obtaining. And so we are testing this null hypothesis that the mean of one group is different from the mean of the other group. That's all we're trying to assess. We can't use z-tests or standard scores to test whether this is a difference because we don't have population information. So what do? Well, William Gossett developed something called the t-test to be able to determine, is there a difference between 2 samples? It follows the exact same logic as a z-test or standard scores test, except what varies is some of the metrics that we use to evaluate it. This is just an aside. Gossett actually developed his testing as part of the Guinness Brewery. He published all his work underneath pseudonym student because he was basically saying, okay, how can I make sure that these different batches of beer are all the same? And so he developed a technique to compare sample to sample to sample over time. He couldn't publish it underneath his own name because he worked for the Guinness Brewery. So he published them underneath that pseudonym student, and that's why we actually call it a student's t-test. Generally that's the term that was used for that. So it's kind of interesting fact about how t-tests develop. So why does the t-test work if we're just looking at samples? Well, the larger the samples, the less likely the difference between the two means was created by sampling error. So if there's a bigger difference, if we have a large sample in each group, the less likely that we're basing things on error. The larger the difference between the two means, the less likely the difference is created by sampling error. So if you have two means that are really close, it might be sampling error. If they're really far apart, it's less likely to do that. And then variance, so that more squished distributions the smaller the variance among the subjects, the less likely that the difference between the two means is created by sampling error. So a really spread out distribution has a lot of variability. That variability leads to sampling error when you and analyze it. So these are things that are under our control. And with a t-test, we actually try to assess these things and controlling for all these factors as best we can. There are two types of t-tests: independent t-tests, dependent t-tests. And both the examples we covered earlier were independent examples. And in an independent sample, you have two different groups that are not connected at all, making up each sample, right? So two groups not related to each other, so they're not affecting each other's results. Dependent samples have two groups that are connected in some way. Brothers in one group, sisters in another, a pre and a post, husbands and wives. There's a connection that makes the people related, not familial, but related somehow between the two groups. You can even do matching. You can say, I'm going to match people on their education levels and their ethnicity and their intelligence, and match people for that. In the dependent sample, the two groups are correlated because the brothers and sisters or husbands and wives or yourself over time, pre and post, should be more similar than if you used a random sample of individuals. So this is a third example. You have a study of visual acuity. You have same-sex siblings. They were identified for the study. Each pair of siblings, there's a coin toss to determine if they're going to receive the vitamin supplement or receive a placebo. As such, the control group is the subject who is the same-sex sibling to each subject. So you have same-sex sibling that is exactly this, is more similar to you than something else. And as a result, subjects are less subject to error than if they were just randomly selected to be part of that study. Matching in this example assures us that the two groups are more similar than if they just were two independent samples used. And that's where the dependent t-test comes in, because it reduces error. You're more similar to yourself. You're a better control for yourself than someone else is, because you have all those same qualities that exist within you. One of the most important factors in conducting a t-test is determining, and I can't see what that actually says, I need to move that, is determining whether you should use an independent or dependent t-test. So you have to make that decision. The questions you ask yourself: are the samples paired in another way? Is it the same person? Is there a connection between person A and person B in these two different samples? Or are the two samples merely obtained by random? So a random sample of boys and girls, a random sample of anybody in one group and a random sample of everybody in the second group. Those are the questions that you end up asking yourself when you're looking at an independent versus dependent t-test. And I can't get that to go forward. I'm going to skip that. So, reporting t-tests. After you have tested whether two samples are significantly different, and we'll get to that near the end, there'll be a lecture part on using SPSS for this. And so to keep things consistent, we tend to report the findings of the t-test by reporting the means, the standard deviations, and the sample size. And we do that pretty regularly for doing that. So you can do this in a table like this. You have group A and group B. You have the degree t-test that you find, so the t-value that you obtained, the degrees of freedom, and then your means. You're writing down your means, you're writing down your standard deviations, and you're writing down your sample sizes. And then down here, it tells you whether it's significant or not. Thing is really just in a bad spot. I got a little floaty thing that floats around that connects to the Zoom, and it keeps getting in the way. So there's one asterisk. One asterisk means that it's significant at the 0.05 level. With SPSS, I usually say just report the actual p-value. That's one way to do it. You could report the t-test and degrees of freedom that was used to evaluate that statistic, including a statement of whether the results are statistically significant or not. So you might say something like, the differences between the means is statistically significant. You report your t, your degrees of freedom, and then your p-value. The null hypothesis was rejected is another way to say that. The difference between the means is significant. There's a lot of different ways to do it. In all cases, the important information is the t, the degrees of freedom, and your p-value. I would say that the most common way to report a t is this format here. T, degrees of freedom, equals whatever the t-value actually is obtained. As a good rule of thumb, when you use the word significant, you should modify it with the term statistically significant. And this is important because statistical significance does not always result in practical significance or importance. And so the example I always give, well, I might find a new study technique that will reduce the amount of time it takes you to prepare your weekly assignments, but it takes me 2 weeks to demonstrate that procedure. Yes, it's statistically significant, but it's impractical. So it's statistically significant, it's not real world significant. And you have to be careful when you're doing that, making those assumptions. And that's why I like to see that word statistically significant. When reporting results that are not significant, you can state very similar things. T, degrees of freedom, your value, and then the P is greater than, or the P is equal to, or that it's not significant, or give the actual P value. In either case, you're again stating that it's not statistically significant. All right, so we're at a break. We're going to take a break. I'm going to stop. I think class is about to start here in a few minutes, so I may go ahead and get that queued up, and we will continue on with the lecture in a few minutes. So I'm going to stop that recording and get it posted.